we can move to our next speaker, Dr. Lin from USA. He will talk about Sparse estimation of correlations among microbiomes. Now you can share it.
Thank you, Professor Nadia. So let me share my screen real quick. Okay, can you let me know if you can see the screen well and hear my voice well?
Yes, WS.
Okay.
So we can see. Go ahead.
All right. Okay, so maybe let's get started. All right, good morning, good afternoon, good evening to everybody. My name is Huang. I am the postdoctoral visiting fellow from Biostatistics and Bioinformatics branch of NICHD. So it is my great honor to be invited and give a talk on our recent methodological work here. So our recent study is focusing on estimating correlations of microbiomes, and the method itself is called the sparse estimation of correlations among microbiomes, or we can call it SITCOM. So the presentation today is about a statistical model, but I promise you there will not be many formulas. Okay, let's get started. First of all, what is microbiome and why are we interested in it? Microbiome are the genes associated with the microbiota, including bacteria, archaea, fungi, and so on. A lot of studies have shown a variety of health outcomes, such as obesity, irritable bowel disease, HIV/AIDS, are possibly influenced or even caused by microbiome. And as compared to human cells, we have 10 times more of microbial cells. As compared to 20,000 human genes, we have 2 to 20 million microbial genes. So there's indeed an elephant in the gut. Okay, now we know the importance of studying microbiome. Then what do microbiome data look like? So I will show you a simple example of how this data is being generated. First of all, we have our targets, the ecosystem of interest, such as gut. And we draw a random sample out of it, for example, such as a fecal sample. And then we do the bioinformatic processing, such as the 16S rRNA gene sequencing. And we can get the read counts for each operational taxonomic unit, OTU, or we can call it amplicon sequencing variant, ASV. The OTUs and ASVs can be further aggregated at different taxonomic levels, such as species, family, genus, and so on, based on your own research interest. So at the end of the day, we have the microbial abundance table. So each column represents samples and each rows represent taxon or different kind of microbes. And if we sum across the rows, we have the column total, which we denote as the library size. So please remember this one. This concept has a huge impact on the microbial data biases. So we will visit this concept very soon. Now we have the microbial abundance table. There are actually some statistical challenges of analyzing such data. First of all, it is well known that microbiome data, together with other high-throughput sequencing data, are actually compositional data. What does that mean? It means that observed abundances are relative quantities constrained by the library sizes. So if we go back to the microbial abundance table, you will see all these values contained in this table are constrained by their column total, which is the library size. So these values are not free floating in the Euclidean space. Another important feature or the challenge of analyzing microbiome data is the experimental bias. This bias is ubiquitous. So it exists in every single step of the bioinformatic processing, such as DNA extraction, PCR amplification, and so on. And also importantly, the microbiome data is a typical sparse data. It means that as many as or up to 80% of the entries in the abundance table can be just zeros, so it's very sparse. So keep in mind these challenges of analyzing microbiome data. Now we switch the gear to our main part of this presentation today, which is the correlation analysis for the microbiome data. The paper is under preparation and is about to submit. As we know, in any microbial ecosystem, microbes maintain complex interactions with each other for the growth and stability of the whole system. So these interactions including mutualism, commensalism, parasitism. So I use a very simple figure to show this relationship. So mutualism is like the relationship between bee and flowers where they help or benefit from each other. And commensalism is like the relationship between a pilot fish and shark. So where one benefits while the others are not affected. And of course, the human beings and mosquitoes are a real representation of the relationship of the parasitism. And researchers have found that changes in the interactions among microbiota are associated with various health outcomes. For example, here, two species in Lactobacillus, that is the Lactobacillus iners and Lactobacillus crispetus, exhibit opposite association with preterm birth. The former one has positive association, and the latter one has negative association. So we need to figure out what's happening in these interactions among microbiota. So the correlation analysis is an important starting point. However, as you can imagine, due to the unique features of the microbiome data as we introduced earlier, the compositional nullity, experimental bias, and excess zeros make the estimation of the correlations a non-trivial problem. Additionally, most taxa are not correlated, which means the correlation matrix, the correlation coefficients we estimated from the data should be a sparse one, means most of the correlation coefficients are actually zeros. Last but not least, not all interactions among microbiome are linear. They may have very complicated nonlinear relationship with each other. So these are the gap in the current literature to address these unique problems of estimating correlations among microbiota. So I will show you using a very simple simulation example. So here we simulate a few taxa, and most of them are not correlated. As you can see here, the scatter plots, most of the dots are distributed randomly between two taxons. However, we make taxon 1 and 3, T1 and T3, are perfectly linearly correlated, while we make taxon 2 and 4 perfectly nonlinearly correlated. All you can see here is a quadratic shape. But keep in mind, this is the underlying truth. We usually cannot observe that in our real life. In the real data, after doing the sequencing and after being exposed to all sources of biases, these are the observed abundance look like. As you can see, most of the relationship of microbiomes are distorted in the observed data, and most of the non-correlated taxa now present somehow the positive association here in the observed data. Again, we can only make inference using observed data in real life, and we cannot observe these underlying truths at all. So how about we just apply the very frequently used Pearson or Spearman correlation to these observed data? What will it look like? Okay, now we just apply the Spearman correlation to the observed data. As a reminder, the underlying truth will only have T1 and T3 and T2 and T4 are related with each other. The remaining pairs are not correlated at all because they just random noise. Applying Spearman correlations, we are able to spot the true positive between T1 and T3. We color in green. However, we miss a target here, which is the nonlinear relationship between T2 and T4. So we have a false negative color in yellow. What's even worse is that we falsely claim a lot of false positive here by using the Spearman correlation. As you remember, the observed abundances are actually presenting a sort of like a positive trend in their relationships. So the Spearman and Pearson correlation, which are designed for testing linear relationship, claims a lot of false positive. That's not what we want to see. And then the remaining cells colored in gray are actually true negative. Recent years have seen a development in a lot of tools for estimating correlations in compositional data. One typical example or typical method is called Sparse CC. Again, the underlying truth is that we only have two pairs of taxa that are correlated. So T1 and T3 and T2 and T4. As compared to Spearman correlation, Sparse CC, the compositional robustity methods, can avoid claiming a lot of false positive here. As you can see, we don't have cells colored in red now. And you have a true positive of the linear relationship between T1 and T3. However, these methods are not designed for detecting nonlinear relationships. So we also still have a false negative color in yellow here for Sparse CC. So this is the current gap in the literature. So the current methods including a standard Pearson-Spearman correlation coefficient and Sparse and other methods, can only test for linearity in relationship. Luckily, we have a new concept called distant correlation. This concept allows us to test for nonlinearity relationship. So the main idea of our methodology called CCOM is trying to generalize the concept of distance correlation for microbiome data while addressing for different biases. The bias is either due to the compositionality, the bias is due to the experimental bias, or the sparsity issue. So if we apply our method to this simple simulation data sets, we can see not only we can avoid claiming false positive here, there's no cells color in red, and we also successfully claim the true positive for either the linear relationship and the non-linear relationship. All right, so this is the simple illustration example using the simulated data set. In the last section, I would like to illustrate how to implement CCOM using a real data set. Now we use a publicly available global gut microbiota data. This cross-cultural infant gut microbiome data contains more than 10,000 OTUs from subjects in 3 countries, US, Malawi, and Venezuela. For simplicity, we subset the data to infants we define as age less than 2 years old for the analysis. And the OTU data were further aggregated into the species level for analysis, and the visualization was based on the top 20 most abundant species. Okay, now this is the result. This is a correlation network for these data sets. The correlation coefficients ranges from -1 to 1 as usual, and colored by blue to red respectively. So the more red the color, the more positive association between the pair of species. And the edges colored in green are actually corresponding to the case where correlation coefficient equals to 0. As you can see here, we see a lot of pairs showing the positive correlation in this infant gut. What's interesting is that we observe a cluster of species which belongs to genus Bifidobacterium are showing a strong positive correlation and cluster together. And this genus Bifidobacterium is commonly available in breast milk. Another interesting cluster we identify is this one. And these species are all anaerobic bacteria, and they are clustered together using the strong positive coefficients. And these 2 clusters may be an indicator for mutualism or commensalism during the growth of the baby's gut. Okay, as a summary, analyzing microbiome data is a non-trivial problem because of the unique features of the microbiome data. So these features including compositionality, experimental bias, and sparsity. Our methodology CCOM corrects bias due to these features and leads to an unbiased correlation analysis. What's important is that the methodology can not only identify linear correlations, but also detect nonlinear correlations. And so it fills an important gap in the current literature. So the work is done with my supervisor, Dr. Shamil Das-Padada, the branch chief of the Biostatistics and Bioinformatics branch of NICHD. And thank you so much for listening, and I'm happy to take any questions.
Thank you. Thank you for your amazing presentation, your organization, your way in giving this speech.
Thank you.
Okay. Thank you.
So I have a question. I don't know if you can hear me.
Sure, I can hear you. Mm-hmm.
Okay, can't see me, but you can hear me. Great. So that was indeed an outstanding talk. And as somebody who has difficulty following these kinds of analytical processes, I think I get it. You certainly made it very clear, so I appreciate that. So my question to you is, you've focused here using CCOM and the strategy, looking at the microbiome and microbiota, and looking at distance correlation between colonization. I'm wondering how applicable this particular strategy would be for those of us who work in trying to understand signaling networks and how there's crosstalk amongst networks, and there may be distance correlations there. So instead of populating these fields with bacteria or pathogens, can you do the same by populating where you see, for instance, different transcription factors, which would network for different signaling pathways? Could you use the same kind of strategy? Because very often we do get false, correlations or false positive, false negatives. Do you see an application there as well?
Yes, I think that is a great question. So although I just organized my talk focusing on the correlation, discovering correlations among microbiomes, but actually the method is not restricted to the microbiome data itself, because the idea is the same. Like other high throughput sequencing data, they have the same similar issues of microbiome data, such as the compositional nullity, the experimental errors and sparsity and so on. So the strategy could be pretty much the same. Like we address the errors, like the biases first, and then we do an unbiased inference of the correlation analysis for these data sets. And I think your question is more like whether we can apply this methodology to other omics data or aggregating multi-omics data together. And the answer is yes. And that is one application of our method. We can aggregate multi-omics data together, but it's a little bit more complicated because we have to address the bias, some bias unique to sort of by a different data type. For example, the metabolomic data, they have a very different data type than the microbiome data. We have to address it carefully for the bias unique to that feature. So that at the end of the day, we can have some a very giant network which connects not only just the microbiome data, but also different data types together. Yeah, that's one of the application and also one of the future directions our method development. Yeah.
And presumably one of the key features here is that if there are little data, there's a paucity of data. When you showed your proof of principle, it was with very small numbers relatively, right? So the advantage here is that the numbers, your N, need not be very high to be able to do this kind of these kinds of distance correlations. Is that an assumption I've made that's correct?
You mean when we say the numbers, do you mean the sample size or?
Yeah, sample size. Yeah.
Actually, yes and no. I have prepared a very mathematical slides here to show the idea.
Yeah.
So the main result of our method is this one. So this is the main theory. So the consistency results. So the R now here, we use that to denote the true correlation matrix. And R hat is the correlation matrix returned by CCOM in our methodology. So here, the consistency theory indicates that so long as the sample size gets to infinity and the number of taxa are not exponentially larger than the sample size, we have a consistent result as the underlying truth. So, so to your question, I think on one hand, we would like to see the sample size to be large. So that, statisticians always like large sample size, right? So if we have large sample size, and on the other hand, the number of taxa, the number of microbes are not extremely large or exponentially large than the sample size, we can get a satisfactory result using our methods. So this is the main theory.
Yeah, so many omics, like transcriptomics, you have huge numbers of data, but your N might be relatively small. So what you're saying, this is doable.
Right, it's doable. And what is interesting in the microbiome data, you are right. For example, like the gene expression data, we have 10,000s or even 100,000 of genes. But luckily in the microbiome data, we can always aggregate these. So for example, we may end up with 10,000 OTUs, but we can aggregate OTUs to species, to genus, to families. So we can reduce the number of taxa here by the phylogenetic tree. So this is the lucky part of microbiome data. We can always aggregate that based on our research interest.
Okay, so I think you can expect that you're going to be swamped with people asking you to analyze their data. So watch out. All right, thank you very much for me.
Yeah, thank you.
Is there any question? Graham?
No, it was a really great talk, very interesting. Thank you so much.
Thank you.